The goals of the experiment were to identify the direct protein-protein interactions of calcium channels in yeast, to compare the morphology and electrophysiology of neurons with functional RIM deletions with control neurons, to determine their role in neurotransmitter release, to observe effects of the RIM deletions on calcium influx and calcium-dependent release, to determine which regions of RIM are responsible for binding calcium channels and synaptic vesicles, to identify the domain of RIM responsible for localizing voltage-gating calcium channels to the active zone using rescue sequences, and to compare calcium channel immunofluorescence in neurons with RIM deletions and their controls to further support the above. Rat protein calcium channels protein interactions were analyzed in yeast. Calcium channels were shown to have a direct protein-protein interaction with RIM and RIM-BP. The direct calcium channel interaction with RIM was unexpected and further analyzed. The C-terminus of calcium channels that control calcium influx presynaptically were shown to have a direct interaction with the PDZ domains of RIM. This was tested by observing the protein-protein interaction in yeast. In summary, it was determined that C-terminus RIM domain, PDZ, is the only domain that binds calcium channels. Spectroscopy analysis demonstrated that calcium channels sequences fits well into the PDZ binding pocket.
The function of RIM was determined by using reverse genetics in mice. Genetically modified mice were obtained by creating genetically engineered embryonic stem cells. The stem cells were then used to create genetically engineered mice whose mutated neurons could be isolated in vitro. The neurons isolated from these cells could then be triggered to delete an essential exon from the DNA sequence. So the goal is to delete a region essential to protein function so the role of RIM in the active zone can be better understood. The genetic modification involved the insertion of specialized DNA sequences on either side of exon 26. These could be triggered at a specific time so that the DNA between the two sites was removed or deleted. So in our diagram here, we see the specialized DNA inserts in red and exon 26. Exon 26 is the first exon shared by all RIM isoforms. Therefore, it was chosen to be the functionally essential domain, which it did turn out to be. Once the neurons were modified, exon 26 deletion was triggered to generate neurons with RIM deletions or not triggered to generate the control neurons. These two groups of neurons were then used in several experimental comparisons. First, morphologically. Once neurons with exon 26 deletions were isolated in culture, the synapses of control and knockout neurons were compared using antibody staining. In our representation, because the postsynaptic density was the same, we are only showing the presynaptic densities. As you can see in the representation, the neurons were equal in synapse size and density. However, when the neurons were also compared using electron microscopy, it was observed that the neurons with the RIM deletion had a twofold decrease in the number of docked synaptic vesicles. This data strongly supports the role for RIM proteins in vesicle docking. Then the neurons were compared electrophysiologically. Observation of the effect of the RIM deletion on neurotransmitter release provided insight into RIM's role in the active zone. The neurons with the RIM deletion were then electrophysiologically examined in order to determine the severity of the deletion on the neurotransmitter release. Observation of the excitatory and inhibitory postsynaptic responses evoked by the control and RIM deletion neurons were compared. It was determined that the RIM deletion caused the amplitude of the postsynaptic, excitatory, or inhibitory current to decrease. Observations also showed that the frequency of spontaneous minis also decreased in the RIM deletion neurons. More importantly, these electrophysiological comparisons showed that loss of RIM function in the neurons caused an increased rise time and rise time variability during an inhibitory postsynaptic current, meaning that the release of neurotransmitter at the presynaptic neuron was decreased and desynchronized, as the demonstration will now show. First, the control neuron. And now the neuron with the rim deletion. As you can see, fast and synchronous signal transduction requires the presence of rim, meaning rim has another role besides vesicle protein. Based on this data and the evidence that rims directly bind calcium channels, it was hypothesized that rim proteins bind and tether calcium channels to the active zone in order to increase the efficiency, speed, and synchrony of neurotransmitter release. In summary, the function of rim in the active zone was impaired by the deletion of exon 26. The deletion had two major effects. 1. Fewer synaptic vesicles were docked in the active zone. And 2. Neurotransmitter release was decelerated and desynchronized.